Hello everyone, so my name is Banerjee and I welcome you all to the very first video of this particular channel. The name of the channel is Bio-ProTech, as you must have seen in the video thumbnail. Okay, so as I have already stated that this is the very first video of this particular channel, so shower some love. Okay, and don't forget to hit the like button and comment down below. Okay, so my name is Banerjee and I am your host, and I am a bioinformatics and AI scientist at a Bangalore based company, and I have decided to start this particular channel to help out all of you out there to kickstart your career in this particular field like bioinformatics, AI. Okay, so I have some 7 or 8 odd years of experience from some of the pioneers in this particular industry like IIT Madras, AIMS Delhi, okay, ICMR, CSIR, and likewise. Okay, so I would try to transfers my knowledge to all of you. Okay, so if you are willing to learn this particular subject, so come along with me. Okay, so we will discuss in this particular channel, so because it is an introductory video, so let me tell you what we'll be dealing with in this particular channel. So first and foremost, obviously we'll be talking about all the life science, biotech, okay, bioinformatics, AI based stuff. Okay, so how computer can be integrated with your biological knowledge, or basically nowadays the biological experiments are generating a whole lot of data. So how to manage that particular data? There comes the bioinformatics, there comes the AI into the picture. So we will try to use the computer to analyze the data, because as I have already stated that there is a whole lot of data, then we want to predict. Okay, so let's suppose the prediction of the protein structure that we'll be dealing with in today's particular first video. Okay, apart from that, your computer aided drug designing and discovery, that is my main domain expertise. Okay, so apart from that, like in this particular channel we'll be discussing about your drug discovery, then your all kind of different tools that come in, very latest tools of artificial intelligence for your protein prediction, then your all kind of multi-omics analysis as well, starting from your genomics, then your transcriptomics, then your proteomics, metabolomic analysis. Okay, so all of those things. Okay, so apart from that, all of the things that we see in day to day or we encounter, like the next gen sequencing experiments. Apart from that, in drug discovery we need to model a new protein, or we want to come up with a new protein structure. Okay, by using some of the classic tools of bioinformatics like your Swiss model, okay, or your I-tasser for your Ab-Initio modeling. We do homology modeling using Phi-Ray or Swiss model. Apart from that, we have your AIML based tool for the same, like your AlphaFold is there, then you have boltz too. Okay, so we'll be discussing about all of those in today's video. Okay, so but apart from that, stay tuned to this particular channel, so we'll be coming up with more of such demonstration videos, okay, in the upcoming future. Okay, so that is there. So in today's video we'll be dealing with protein modeling. Okay, so without further ado, so let's get started. But just before getting started, I just have a suggestion to ask from all of you, from all the viewers, because this is the very first video of this particular channel. I just want to request you that can you please comment down below that what would be the preferred language in which you want to have the content in. Okay, so because I haven't fixed any of the language as of now, but obviously I'm speaking English as of now, but I can do like Hindi also, so I can do English also, I can speak Bengali also, Spanish also. Okay, so but I can also do bilingual, English, that is Hindi and English both. Okay, so some part of English, some part of Hindi, so that is obviously possible. Okay, but just to keep the things professional in the first video, so I have just chosen English as of now. But if you all comment down below as per that particular suggestion, I will try to make the further videos as per your suggestions. Okay, so that is there. So I will just pin down the poll if possible in the comments, or you can just directly comment in there. Okay, so yeah, that is there. Okay, so without further ado, so let's get started with the demonstration. So I will be presenting my screen and I will show you about the 4 tools that are there. Okay, so the first tool is your Swiss model, which uses homology modeling. Okay, then we'll head on to Ab-Initio Modelling. For that we'll be utilizing the tool called as I-tasser. Okay, then we will move on to the AI/ML based tool. Okay, so that is a MIT based server. So Massachusetts Institute of Technology provide that particular server for free for all of the academic people or research based people to use that. Okay, so obviously it is a open source software. Okay, and it is powered by NVIDIA. So in the background, supercomputers and the quantum computers even enabled with GPUs and NPUs are being working in that. Okay, and we can leverage the use of that particular thing for free. Okay, so that is called as your like NVIDIA based server. Okay, so we will look into that as well. Okay, so we'll look into three major tools. Okay, and within the third tool we'll look into two different kinds of like softwares out there. So one is your AlphaFold and the next one is boltz. So while we discuss, I will also explain it side by side that why it is used. Okay, so as much as it is possible, as I cannot stretch the video like too much. Okay, so it would be like it might get boring, so that is why I have decided so it would be 30-40 minutes video. So in that particular time period, how much we can justify the topic, we will try to do that. Okay, so yeah, so let's get started with it. Okay, so first things first, so all of the tools we are going to demonstrate in today's session are going to be free to use. So basically they are freewares. So freewares are tool which can be utilized for free or basically used for free. So you don't have to pay any penny to access them. Okay, why we are using such tools or basically web servers? Because obviously so that maximum of you can use it okay, without any money involved in that. Apart from that, it also surpasses the barrier of your OS, okay, operating system. Basically some of you might be utilizing your Mac OS, or someone of you might be using your Windows or Linux, okay, or Ubuntu and likewise. Okay, so or any subsystem of Linux also, or cloud servers. Okay, so that is why so there is no barrier to it. So you don't have to install any kind of software. Okay, so all of the tools that we are going to demonstrate, we are gonna do it in web. Okay, so so all of the websites are having their own servers, okay, cloud servers. So like all the analysis, be it your like supercomputer based search, okay, or GPU based search, everything will be done by them in the background. So your computational power is also not required. Okay, so any of the computer you can use for the same. Okay, so irrespective of its CPU GPU configuration. Okay, because everything is going in their servers, mother servers. Okay, so we are just gonna utilize or leverage the use of that using the web server. Okay, or using simple internet. Okay, so you can simply go to your regular search engine. Here we are using Google. Most of you might be using Google itself. Okay, so the first tool that we are gonna see in today's demonstration is Swiss model. Okay, so as you can see from my screen, this is Swiss model. Okay, and the Swiss model is from a company based out of Switzerland called as the Swiss Institute of Bioinformatics. Okay, so this is a very big institute based out of your Switzerland, and they have plethora of tools like concerning to bioinformatics, drug discovery, protein modeling. Okay, and likewise. Okay, so we will straight away get on to start modeling. So first and foremost, we are like diving deep into modeling and all. So what is modeling? Okay, so modeling is basically initially you will be having a protein structure. Okay, so protein structure is a bits in a string kind of a thing. So all the amino acid would be stacked and you have a sequence of amino acids. Okay, but that is just the primary structure. When it is gonna fold in the secondary or your tertiary or quaternary structure, so it is further more complex by the help of your disulfide bonds and peptide bonds. Okay, so it will acquire some structure. Okay, so it will be having some contours. Okay, some solvent accessible surfaces. Okay, so how it is gonna fold, that we are gonna determine, and that is very important. Why is it important? Because the structure of the protein gives it the stability. The backbone of the protein structure gives stability to the protein, and that obviously vouches for the quality of the protein as well as the functionality of the protein. So if anything happens at the structure level, okay, so it will be reciprocated at the functionality level. So if any malfunction happens, okay, or there is some, let's suppose, proline or lysine, some kind of amino acid which are very flexible, so which are basically destabilizing at some point in time. Okay, so that can have some different effects, like the variances. Okay, so let's suppose we are talking about a mutant. Okay, so how a mutant is different from a wild type. Okay, so it might be having several of differences or one or two amino acid changes. Okay, so that particular thing brings about a change in the structure of the protein and thereafter changing the functionality of the protein. That is what we are concerned about. Okay, because most of the disorder that we think of are mostly caused by the variance can be caused at a genomic level also, but we are concerned at the proteomic level. What are the things happening? Okay, so we can identify that particular variance either at genomic level. For identifying that at genomic level, you can simply go to NCBI. Okay, so NCBI you can go on to. Okay, and for identifying the genomic variances and SNPs, you can simply go to your SNP. Okay, then you can search by the gene term. Let's suppose PNPLA3 that causes your non-alcoholic fatty liver disease, and those things we can search for. Okay, and likewise, that is just an example. Okay, so let's suppose we are continuing with that particular example, PNPLA3. Okay, so so you will be finding all the variances, okay, in the form of SNPs or SNPs. Okay, so you can see what a single nucleotide variance, okay, what kind of variance at what particular what is the reference allele, what is the alternate allele, everything will be given over here. But mostly we are here itself we are doing protein modeling. We are not concerned about the gene particularly. So you can simply go to PubMed to read the paper of the same, that let's suppose I'm taking a particular protein. Let's suppose I'm going to PDB, protein data bank, that is a primary database for your structure search. Okay, so let's suppose I am trying to search for a main protein of SARS-CoV-2 or basically COVID-19. So that is 3CL Pro. Okay, so 3CL Pro. So you can see the first result that you get. Okay, so we are getting two proteins basically. Okay, so this particular protein structure may be procured from a wet lab experiment or a dry lab. Here you can see at the top here, like I'm pointing my cursor. So here you can see like 240,000 odd proteins are being procured using wet lab experiment. Here you can see wet lab experiment, whereas using computational model, particularly your AlphaFold and all. Okay, so we have come up with more than 10 lakh structurally modeled compounds, okay, or basically proteins. Okay, so here you can see the first one. Okay, so 9HFX is there. Okay, apart from that you also have other 9HFY. Okay, so similarly you can get many. Okay, so depending upon what kind of protein structure you are concerned about. Okay, let's suppose I go on to this. Okay, so this is the protein structure of your main protease of your SARS-CoV-2. Okay, so here we can see the classification of it. So you can also read the paper. This is the paper. Okay, so PDB DOI, the paper link is also given over here. What is the method they have employed to procure this particular structure? That is X-ray diffraction. Okay, and here you can see the resolution is also very good, 1.96 angstrom. Anything below 2 angstrom is considered good, and as low this number is mathematically, okay, so it is a better resolution. Okay, so that we can see. Okay, apart from that we have multiple literature, ligands concerning to this that. Okay, so that is a different part altogether. Okay, so in today's session obviously we are dealing with modeling. We will not dive deep into this. Okay, so here why we have come here, we have basically the structure. We have to take the structure from PDB. Okay, either you can download the structure in the PDB form or CIF form, or you can also do it in your FASTA sequence format. Okay, or here it's here right now we don't require to download it. Okay, so you can download, that is not a problem. You can simply you can simply go to display files and you can see the FASTA format, because you just have to copy, so no need to download. Okay, so we can simply copy this particular sequence. Okay, and then we can do the mutation. So then how to do the mutation? We need to study that where the mutation has happened. For that you can study that via multiple literatures. Okay, you can go to PubMed and then see that what are the reported mutation concerning to this particular protein. That is one way. Another way, if you want to search for it, so you can simply go to OMIM. Okay, OMIM you can go to, that is, you can also go by via NCBI also. NCBI in the drop down menu also you get that. Okay, OMIM. Okay, so yeah, so this is online Mendelian inheritance in men. This is the full form of OMIM. Okay, here you can search by the gene features or clinical relevance features, okay, or like protein and more. So you can search, okay, and then you can come up with what is the particular variance concerning to this particular protein. Okay, so obviously we are not dealing with that, so that will stretch the video for a long time. Okay, so we don't want that. So basically we will search it over here and find the variance, or you can read the paper and find the variance, that where the variance is. Variance, why we are looking for? Because obviously the wild type structure is already available. Modeling the wild type doesn't make sense, it is already available. Okay, so we will do, or basically we'll model a protein structure which is not available. So the mutant of this is not available, okay, mostly because we will be doing mutation. So basically I will be doing an arbitrary mutation. So based on the research paper you can do your mutation. Okay, so I will just show you the way. Okay, I will just show you the path how to do it. Okay, so simply we are not concerned about the header. So this is the FASTA header, basically starts with this particular symbol, then you have your annotation. Okay, so then you have chain, and then what is the sequence is all about. So it is 3C like proteinase, so also called as NSP5, that is non-structural protein 5. Okay, and it belongs to the organism that is written, that is SARS-CoV-2. Okay, so yeah, so now we just have to copy this particular sequence. Okay, Command C I am using because I am using a Mac. You can also do Control C, or you can simply right click over here and copy. Okay, so whatever is feasible for you. Okay, so now I will go to notes. So if you are using a Windows, you can simply paste it in your Notepad, Notepad Plus, or your Word also. Okay, our simple task is we need to do the mutation. For that you can also use text editor for the same. Okay, let's suppose, as I am already stating, that I am doing a arbitrary. Okay, arbitrary, obviously I don't know actually, so I am doing a arbitrary mutation just for you, for your ease to show that how it can be done. Because it is a demonstration, we are just taking an example. So you can see at the 4th place we have a R. Okay, so you can zoom in, okay, and see that we have a R over here, and I will replace it with F. So this is the variance. Okay, instead of R in the 4th place, we have replaced it with F. Okay, so obviously that is a mutation, it becomes a mutation. Okay, so you can do multiple mutation, but at once don't do. Okay, one by one you go. So now this becomes a mutant sequence, because at the 4th place in the wild type, what you were having R, and now you are having F. Okay, so again do the same thing, copy this new sequence, and because we are concerned with like modeling, okay, so we will simply paste it in the Swiss model. Okay, so we'll simply paste it over here. Okay, here you see it is pasted, and now we will go to search for templates. Okay, so before searching for templates, let's understand what we are trying to do over here. Okay, why we are using Swiss model, what does the template means? Basically Swiss model is used for your homology modeling. Okay, so what is homology modeling? So there are multiple types of modeling, particularly your homology modeling is there, then you have your ab initio, then you have your threading. Okay, so in homology modeling is having most amount of your significance and most amount of confidence we are having. The reason behind that is we are searching for the homologous sequence, or basically homology, or you can say in simple words, we are searching for the ancestry of it. Okay, so this, let's suppose this is a mutant sequence. So now it has become a mutant sequence. This is called as your query sequence. Okay, now if I try to find a sequence, or like in other words it can also be called as your target sequence. Okay, so in query sequence or target sequence, so this particular sequence I want to match that. See, this particular structure is not available. Okay, so I am doing it like a novel protein modeling or structuring I am doing, but a similar protein structure might be available, no? So what is similar? Obviously the wild type would be the first similar, because we have just replaced it with only one amino acid. So it will resemble with the wild type 98 to 99% even. Okay, so approximately that. Okay, so apart from that, so what it will do a multiple sequence alignment. Okay, so this software, what it is doing, it will initially do a multiple sequence alignment and try to find the match of this particular sequence to all of the protein that are available in this world. Okay, so basically in the PDB, it will search against PDB. This particular sequence will be searched against PDB using all of the supercomputer. Obviously, they are having supercomputers in the background, which is carrying out the multiple sequence alignment. Okay, and when it will find that anything which is having a sequence similarity of more than 33.8%, that can be called as a homologous sequence or template. Okay, same thing. Okay, so it is just same as how you have a reference genome, no, to which you compare. Similarly for protein it is called as a template. Okay, or wild type also you can call it as, but template is basically how much resemblance it is having. For homology modeling, generally it is said that also it is a norm, I will show you. Okay, so in the documentation section you can see basically. So that is 50% and more. Okay, 50% of resemblance or 50% of sequence similarity we are looking for. Okay, so how to get that? So anything beyond 50%, let's suppose it is able to find 1000. Okay, so I will search for template, and in the meanwhile I will be explaining. Okay, so because it will take some time, so here you can see it is already running. Okay, it is searching for the template. Okay, so in the meanwhile, while it is queuing and the process is running in the background, here you can see all the codes over here. Okay, so you can see my cursor. Okay, so here it has start running. Okay, so now it is searching this particular sequence, trying to align this particular target sequence of ours to all the protein sequences and structure that are available in PDB. Okay, now let's suppose it comes up with 1200 odd sequences. Okay, still it will give you top 50 based on GMQE, Q-mean and Z-score. Those are the evaluationary parameters. Basically, we'll talk more about that while the result comes in. Okay, but basically GMQE and Q-mean, mostly we are looking for more in the 1, the percentage that we have over here is 0 to 1. 0 means 0%, 1 means 100%, because it is a probability based value. Okay, so the maximum that we can expect is 1. Okay, so 0 to 1. Okay, so GMQE is there, but although GMQE is your coverage dependent, if the coverage is 100%, then we can expect a maximum of 1. Although 1 we won't be getting, because that is a theoretical limit. Maximum we can expect near to 1. Anything near to 1, 0.91, 0.92 is very good result. Okay, same goes with your GMQE. Okay, so GMQE is closer to 1. Similarly is your Q-mean. Q-mean should also be closer to 1. Q-mean is not related to your coverage, so it doesn't matter. Here you can see it has extracted 1110 templates. Okay, still it will give me top 50, because I cannot screen. Humanly it is not possible to screen, so basically it will give me top 50. Okay, so top 50 based on the sequence similarity. Okay, so here you have the results. Okay, so you can see the results. So the first result you can see GMQE is 1, so that is the maximum. Okay, then QE is again 1. Okay, and the identity is 99.67. Why 99.67? Because it is trying to align it with what is the first result. I haven't given it that this is from SARS-CoV-2 or anything, any kind of knowledge I haven't given, but still it is able to find it. See, the first template is the wild type only. How do I know that? You can see the first template, that is 3C-like proteinase NSP5. It is able to find it by itself. Okay, so because it is very similar to the wild type, only one amino acid we have changed. That is why the identity is 99.67, because only one amino acid we have changed. But this will still influence the structure. It is not that we have changed only one amino acid, that won't influence the structure. Okay, it is not that. Okay, one amino acid change can also influence the structure and also governs its functionality. That is why we are looking at. Okay, mostly we do it for the diseased condition, for a diseased condition, if a mutation has happened, let's suppose JN.1. okay, so the spike protein mutation in the SARS-CoV, we have seen that. Okay, likewise we can model that particular protein, and what is the purpose of that? Obviously you can model that particular protein, then validate that particular protein. Okay, so this is a mutant protein, then we can come up with the therapeutic intervention. So that we will deal with when we have a video on your computer aided drug designing, that how do we design ligand against that. Okay, how we can come up with a novel ligand against your SARS-CoV-2 or any kind of disorder for that matter, any kind of neurodegenerative disorder, cardiovascular disorder or cancer, this that. Okay, likewise. Okay, so yeah, so I can do multiple modeling. Okay, so I can click on them and one by one I can do, or multiple I can do. Okay, so let's take 2 and simultaneously it will model 2 proteins. Okay, so let's hit on built model. Okay, so you can order it based on GMQE, Q-mean or your Z-score. Okay, likewise GMQ is best only. So let's stick to that. Okay, so it is running. Here you can see once it is ready, so the model will appear over here. Okay, because we have done two models simultaneously, so two of the models are showing over here. Okay, so I can click on this one, the second one, or the first one. Okay, so this is the second model that is aligning over here. Because the coverage is like 100%, that is why you can see this is the template, green one, and here is my second model, here is my first model. Likewise, n number of model that you are modeling over here, so it will be like all of them will be stacked over here, so that we can compare it. Okay, apart from that, let's see what is the result all about. Here you can see so let's focus on the second model. This is a very good model. How do I know that? So you can see the coverage. I have already stated that the coverage is 100%. How do I know? So this particular rectangle that you see, if it is fully filled with blue, that means that is 100%. If it is like 90% filled, so that is 90% likewise. Okay, so you can just point your cursor. Okay, so and it will show you that particular thing, how much it is having the coverage. Okay, so we can go to the previous template part and it will show. Okay, let's go to template. Okay, and if I point my cursor towards here, you can see chain 1, range 1, and coverage is 1. 1 means 100%. I have already stated, because these are probability based values. Okay, so now here you can see this is the graph of the Q mean. Okay, and we'll also require one more graph, that is a Z score. Just you point in here. Okay, so this is the Z score graph. Okay, so GMQE should be near about 1, and it is already 0.97, so that is very good. Okay, 0.97, what is vouches for? GMQE stands for your model quality. Okay, so basically model stability also you can see, basically it stands for stability majorly. Okay, so it vouches for the stability of the protein. Okay, Q mean stands for the quality mostly. Okay, that is 0.91, and you can also consider the standard deviation, so it becomes 0.96 if you add this one. So maximum it can go is 0.96. So again it is very close to 1. Okay, so again it is a very good result. So Z score should be, so let's understand this. Okay, so Z score should be so the Z score over here, okay, should be here you can see like if it is in the darker gray area, that means the Z score is between 0 to 1 or less than 1. Okay, and if it is in the lighter gray area, that means it is between 1 to 2. Okay, and if it is anywhere in this particular vicinity of very light gray or white, so it is not a good model. Okay, so we want our red star, that the model star, here you can see a star, red star, so that is lying in the darker gray area, although, but it should basically lie over here. Why is it happening? Because I have done an arbitrary mutation. Okay, if you study the mutation and this is the particular mutation, then it will lie in the very good area only. So if it is lying a bit over here, it is a very good model. Still it is a very good model, because at the end of the day it is lying in the darker gray area, although the model is lying a bit out of the plane. Okay, a bit out of the plane, but that is okayish. Okay, so it is a decent result, you can say. Okay, just for the Z score. Apart from that, GMQE Q mean is very good. Okay, so but we take the cumulative count. Okay, so like Z score should be less than 1. Okay, apart from that, GMQE Q mean should be closer to 1. Okay, so that if all of the rules are being satisfied, that is a very good model. Okay, yeah, so that is there. Okay, so likewise you can also see the first model. Okay, first model is a bit lower than that. Okay, so let's also see the Z score of this. Okay, so Z score is similar to this. Okay, so the previous one. Likewise you can model, you can also download this particular structure. So here our homology modeling is done using Swiss model. Okay, so you can also use an alternative tool for the homology modeling, like Phi-Ray is there. Okay, Phi-Ray is there, or like Swiss model is very widely used. Okay, now let's come on to another type of modeling, that is called as an Ab-Initio. Okay, so here we have talked about if there is 33.8% and more similarity, majorly 50% and more similarity, then we can find the template of that particular a target sequence that we are searching for. Then we can simply do the modeling. But what if I am coming up with a very novel sequence or very novel protein, and now I don't have a template, or basically any of the protein structure in the PDB does not resemble this particular protein that sequence that we have, or the mutant that we have, then what to do? We will not model? No, we will do. Okay, so for that we will be using a particular type of method called as your Ab-Initio modeling. Okay, for that we will use a tool called as I-tasser. Okay, I-tasser. Okay, although I cannot demonstrate this particular tool to you fully because this is a supercomputer server and it generally on an average takes 48 to 72 hours. That means your 2 to 3 days it takes, because it is very complicated way of your modeling. The reason is Ab-Initio means, Ab-Initio is a Greek word meaning from scratch, because as you don't have a template to build your structure upon, so you have to build it from scratch like absolute base. Okay, so what it will be done, so you have the whole amino acid, let's suppose this is the whole amino acid sequence for you, so you will be splitting it into multiple small chunks. Okay, and obviously the whole of the sequence is not matching with any of the templates, so what to do, we can match the small chunks over there, and obviously the small chunks, let's suppose 1 to 8 amino acid, then 9 to 16th amino acid, so that is resembling with some of the proteins. So this particular parts would be individually modeled, and then using physics based refinement we will be averaging out all of those, and all of those low resolution models will be clubbed together to form a protein. Okay, the whole protein will be formed. So as this is a very tedious process, okay, obviously this is taken care of by supercomputers, but obviously following a supercomputer, and obviously it will build each and every bond and everything. Okay, and likewise, so that is why it takes some time. Okay, so here we can just do the same thing. Okay, so same sequence, same mutant sequence, you just have to paste over here. Okay, it is from Zhang lab. Okay, so this is maintained by National University of Singapore. Okay, so this is a very famous university from Singapore. Okay, it is their web server. Okay, called as your I-tasser. And this particular lab, so there is a lab called as Zhang lab. Okay, basically this Zhang lab has multiple tools. Here you can see on your left, okay, you can see multiple types of tools they have. Okay, from your drug designing to your protein modeling, multiple tools. Okay, so you just have to paste your sequence over here. It can accommodate up to or 10,100 10,000 and 1500, 10,000, 1500 you can say. Okay, so or yeah, no, it is not 10,000, pardon. Okay, so it is basically 1,15,000, pardon. Okay, so this is 1,15,000 amino acid sequence it can accommodate. Okay, so in here you can paste that many sequence. It is not that one sequence only, you can paste multiple sequence as well. Okay, and if you have it in the form of a Word file, you can just simply click on choose file and upload it. Okay, so that is there. Only a problem is there with this particular site is it will only take your academic email ID. Okay, so either you are associated with your college, or mostly obviously you'll be in some college or your research institute or something like that. Okay, so it will only take the institutional ID. Okay, so it will not take your personal Gmail ID and likewise. Okay, Gmail, Yahoo, those things it won't take, because it will take, but there you have to subscribe to them and you have to pay some money, or that much that part is paid. Okay, only for the academicians it is free. Okay, so you just have to paste your mail over here and then hit on run I-tasser. Okay, so it will run in the background. Okay, so you obviously don't have to open keep it open. Okay, so it will be submitted to their server, and after 3 days when the results are ready, they will mail you that particular link. Okay, and you can click on that particular link and you can have the results. Okay, so like that. So that is there. Okay, so this is the so you can also cite their journal, so where multiple of the papers are published for Ab-Initio modeling. This is a very widely utilized tool all across the globe. Okay, so last but not the least, now we will be diving deep into some AI/ML based tool. Okay, so let's understand how a AI/ML based tool works in the first place. Okay, artificial intelligence machine learning based tool. Okay, so first artificial intelligence based tool that is very famous, all of you might be knowing about it, is AlphaFold. Okay, just to give you a stat or a trivia, so by the year 2023, all of the protein that is known to humankind, okay, has been modeled using AlphaFold. So that much of an importance it is having. Okay, So because it is like previously it used to take 7 to 8 years to procure a single structure of protein. Now with the advent of an AI, ML and bioinformatics based tools that I am showing, okay, the previous one also you saw that it took only hardly 5 minutes, not even that, okay, and using AI, ML based tools it will take some seconds, okay, seconds to minutes, okay, 1 minute or hardly 1 minute, 2 minutes, okay. So as compared to a 7 year to 2 minutes, that much of a difference we are having, okay. And if you are like questioning about its validation methods or likewise, it is not like that. You can read the paper. So the paper of AlphaFold is published in Nature, okay, you can see this is published in Nature, and the Alpha Missions is published in Science. Obviously these are the most and highly revered journal in this particular field of your biology as a life science as a whole, okay. So obviously this is having 98% or more resemblance. If you can go through this particular paper, if you see this particular part itself, okay, so here only it is written. So you will see that if we superimpose a particular experimentally validated structure that is shown over here in green, and you have a computationally modeled structure, particularly AlphaFold based model structure that is in blue, you can see how much resembling it is, okay. So all of the alpha LSS beta sheets are absolutely superimposing, okay. So and it is having, and it can go up to 98 to 99% of resemblance, okay. It is not that only the outer part of it or the secondary structure, N terminal, C terminal, it is not like that. You can also look at the different metallic ions or basically the active binding domains, okay. So that is also resembling if you just superimpose that. Here you can see green again experimental, AlphaFold based structure, blue, okay. So and the whole protein structure as like the whole protein structure with its backbone as in the side chains, everything you can see it is having very high resemblance. Here you can see RMSD is very low, anything below 2 is very good, okay. So for a domain specific, and for the whole protein 2.2 is also very good result, okay. So here you can see likewise. Obviously we are not doing a 101 on AlphaFold as a whole, just I am trying to state that how the modeling in AlphaFold you can like infer from that, and how you can understand that. We are like basically looking into that particular tangent, okay. So here you have this, okay, so this is your AlphaFold, okay. So AlphaFold based like protein modeling, what we can do, so we can search it via multiple means. We can search it via your name, okay, the organism name, or your accession ID that you have, or the like sequence structure, okay. Let's understand about like obviously here we cannot directly model your mutant protein because this is the user interface part, okay. So here let's suppose we are going for free fatty acid, okay. So here you can see this particular thing is already being modeled, okay. As I have already told you, all the protein known to humankind has been modeled using AlphaFold till the date, okay. Here you can see just why I have opened this particular site is just to show you how to read the PLDDT. PLDDT is called as your pre-residue, okay, pre-residue confidence score, basically pre-residue local confidence score as a whole, okay. So what is that, okay. So if you want to learn more, you can just go to here and learn more, okay. So what is all about it, okay, just to gist it out, I will state. So if we have a PLDDT of more than 90%, so the color connotation that you can see is darker blue, okay. So it will be represented via darker blue. If I just click or basically just point my cursor towards here you can see the PLDDT is 98.01, that is very high. What does it mean, okay. And the lighter blue is between 70 to 90%, okay. The yellow is between 50 to 70%, and then orange is lower than 50%, okay. So what does it signifies? It basically signifies that let's suppose this particular model is done using computer, okay, obviously the AI ML based tool. So if a particular wild type structure is being procured for the same protein, if we like procure this particular protein structure using wet lab experiment, be it your X-ray crystallography, NMR, okay, or cryo EM or EM, okay, electron microscopy, likewise, okay. So what are the odds that how much resemblance we can get? So this particular backbone that you see, which gives the stability to the protein, so it is having very high chances of getting a match. 97% this particular area will exactly match to the wet lab experimentally validated protein, okay. Likewise, So we can validate this particular thing using in silico, also using molecular dynamics simulation and all. Okay, so that is very much possible. Apart from that, if you just compare it to wet lab experiment also, okay, so you can see we are looking for a higher confidence score. Okay, so because what does it signify? I have already told you that this higher confidence scores gives us the confidence that it will resemble the actual wild type structure that is available in the nature. Okay, that is why we are looking that how much close we can get to this, okay, to the wild type structure. So like the more we have the darker blue, the better our structure is in this particular case. Okay, obviously color connotation can change based on the different tools and softwares. You can have different color connotations, but the main thing is a very good structure would be having more of your 90% and above confidence, or at least between 70 to 90% confidence. Okay, so that is what it is shown over here. Okay, so yeah, so time doesn't allow us, otherwise we would have also talked about the alpha missense. Okay, so I just don't want to continue with all of the things in the first video itself, so that is why. Okay, so yeah, that is there, but I'm just giving you the picture of what we are gonna learn in the for the things. Okay, so just giving you a bird's eye view or the holistic overview of all the things that are there. Okay, so yeah, apart from that, some of the parts I have already demonstrated. Okay, so here you have the quite understanding of the AlphaFold, how it is there. Okay, now, okay, so you can read it from the help section. Okay, so all of the paper. So similarly, okay, so just one thing I like forgot to mention. Okay, so if you go to Swiss model also, in Swiss model also you can read about it. Okay, you can go to documentation section, Swiss model. Okay, you have the full publication details alongside how to read about this. Okay, all of the things is being explained over here. Okay, so you can read it very carefully. Apart from that, all the GMQE, Q-mean that I have already told you is being shown over here. GMQ is the first parameter, second is Q-mean, the graph of the third one is the graph of the same, fourth one is the graph of the fifth, that is Z-score. Okay, and model evaluation you can just study. I have already explained all this part. Okay, you can just study, do a self-study in this particular part. Okay, in order to know more about it. Okay, yeah. So because you can explore it in multiple ways possible. Okay, so now coming on to the last and most recent your server that is made public. Okay, that I have already stated, that is it is a MIT, Massachusetts Institute of Technology, based out of US. As you know, it is a very famous institute. They have come up with this server. Okay, and this server is basically powered by GPU's graphical processing unit and NPU's neural processing unit, and powered by NVIDIA particularly. Okay, that is why it is accelerated by your AI. Okay, so and very sophisticated amount of softwares that you get over here. Okay, so we'll just type in MIT. Okay, MIT based server we can type. Okay, or directly instead of MIT we can also type NVIDIA. Okay, NVIDIA. Okay, you can also type NVIDIA based AlphaFold. Okay, Bioinformatics also you can type. Okay, or diff-dock if you want to pursue docking. So we'll also have a separate video on the diff-dock and docking part. Okay, so in the later on videos. Okay, so for now, so NVIDIA we are talking about boltz 2. Okay, so sorry, boltz 2. Okay, yeah, so this is boltz 2. So what is boltz 2? Boltz 2 is basically a very new software. Okay, that is obviously AI/ML based. Okay, and what we are doing in this particular tool is very like fascinating thing we are doing. What is that? In AlphaFold we are able to predict only the protein structures which protein we already know of. Okay, the protein that we know of, we can only model those. Okay, but in boltz 2 there is no restriction of such. Okay, if you give it a sequence, okay, a random sequence, random like amino acid sequence, you arrange it and give it to the boltz, it can still manage to come up with a model structure. So that is the beauty of it. So by this particular tool we can come up with very new models. Okay, so even the name of that we don't know, or even that do not exist in nature, so that is also possible. Okay, so that is the power of your boltz 2. Okay, but already by the default whatever you are having, that is having a ligand and the likewise, so we don't want that. We just simply want to model the protein. Okay, so what to do in that particular case, we simply will be deleting the ligands and all, because we are not interested in that. Okay, so because this particular demonstration deals with your protein modeling. Okay, so we'll just get on to hit on new. Obviously protein is already selected, so we'll just hit on new, and in this particular sequence, protein sequence, I already have it copied, that particular mutant sequence of ours. I will simply paste that, Control V or Command V, whatever system you are using. Okay, based on that, and now I will hit on run. So in the background it is having a supercomputer. Okay, and even nowadays they are planning for a quantum computer. Basically it is powered by that. Okay, it is having multiple GPUs, so it will be so fast. I will just show you. This is the mutant protein model. I am just hitting on run, just see. Okay, once I hit on run, so it will go to generate. Okay, so in this particular generate, it will not take like even a minute mostly. Okay, it depends, although depending upon the sequence, but it mostly doesn't take a huge amount of time. Okay, although if you want to do it in your regular system, forget about that. Even in a supercomputer also it will take months of time. Okay, and this is able to do it in like a few seconds to minutes. Okay, depending upon the sequence. If the sequence is very long, it might take a bit longer time. Okay, and it also depends upon the queue. You can see it was queued. Queue means like obviously it is a super open source server, so I have like lakhs of people, millions of people are using it worldwide. Okay, so that is why. Here you can see we have got a very good model of our mutant. Okay, how do I know this is a very good model? Here you can see this now, that the model is having very high PLDDT score. Okay, so if I just remove this over here. Okay, so yeah, here you can see. So what is the PLDDT? Here the color connotation is a bit different. Like higher, what you say, your confidence score is shown by yellow instead of blue. Okay, so here you can see most of our protein structure is yellow only. Yellow and some part is green. So green is also above 90% only. So 85 to 90% almost. Here you can see. Okay, this is obviously the scale. Okay, so only the side chains that you can see, only the side chains which does not basically gives the strength to the protein, those are having very low like PLDT score. So that is not at all a problem. Okay, so it is a very good score. So we have come up with a very good model. Okay, because we are having very high confidence score. Okay, so this is one way of modeling. Okay, so that is called as your boltz 2. This is the name. Okay, boltz 2. You can try it by yourself using any kind of model that you want to model. Okay, so there are multiple uses of this particular tool, not only this, but I am just showing you for the sake of your protein modeling in this particular video, as you know. Okay, so that is there. Now you can also go for other kinds of modeling. Okay, so it is not that only one type is possible. So we can also use like AlphaFold over here. How to do that? So I will just go to explore. Okay, I will just go to explore, and if I just remove my video to this side. Okay, so I will just hit on explore. Okay, then hit on biology. Okay, yeah, so once you hit on biology, so you have here, you see for the biology what are the tools they have. You have all of these tools for protein structure prediction. You can see boltz 2 is there, ESM fold is there. Okay, MSA search is there, multiple sequence alignment search. Okay, then open fold 2 is there. Okay, AlphaFold 2 is there, AlphaFold to multimer is there. Okay, multimeric protein that you want to model. Okay, apart from that, for virtual screening you have this many tools. Okay, for docking you have your diff doc, and likewise obviously all of them are AI powered. Okay, so artificial intelligence based tool. Then protein designing you have Evo 2 is also there. Okay, RF diffusion is there. Multiple tools, you can, plethora of tools are there. Okay, that they have come up with, and all of or most of them are free for you to use. So that is a very good news. Okay, so you can use this particular thing to pursue your project. Okay, so not only your bachelor's, masters. So I have trained many students from your bachelor's to your postdoc. Okay, so you can leverage this particular tool to kickstart your career. If you are a bioinformatics person, so obviously it is very helpful for you. If you are AI based person, obviously helpful for you. But apart from that, if you want to get started with bioinformatics, you are a biotech graduate or like a postgrad or a PhD, okay, or a postdoc even. Okay, so in that particular case you can get started with this particular thing, and these are of industrial standard. Okay, these are not just for the sake of understanding or learning. These are research scale, okay, industrial standard, and even more than that. Okay, and so likewise. Okay, so we'll just go on to our last tool demonstration for this particular video. This is your AlphaFold 2. I have already demonstrated how to read the AlphaFold part, okay, in the classic AlphaFold tool. Okay, but now we will see in this particular thing that how we can model a mutant. Okay, so same process will just be iterating over here. Instead here we need not to remove anything. We just have to remove the, if there is a receptor, no, there is not. If here the receptor is not there, so easy, much more easier for us. We will just paste the same sequence which we are like pasting from for till now. Okay, that particular sequence are like at the fourth place, F replacing R. Okay, now here I will be selecting SARS-CoV-2 main protease. Okay, because this is 3CL Pro. Okay, I will just simply do this and hit on run. Okay, so I can simply hit on run, because I have already once modeled it, so it has it in the memory. So that is why without even taking a split second it has come up with the result. Here you can see this is the result, and this is the similar result that we have got in the boltz 2 as well, no? Because the sequence is same, so they are gonna model it similarly only. Okay, so if you are giving two different sequences to the different tools, still you can see the comparative analysis that are there any differences or what. Okay, but as I have just now modeled it in boltz 2, okay, and I have previously done this in your AlphaFold 2 as well, that is why it is giving me this particular protein sequence out here. Okay, here you can see the here also we are getting a very good protein structure, because most of it is you're having higher, what is a confidence score or PLTDT. Okay, so yeah, that is there. Okay, so that is all from my side as of today. Okay, so if you intend to find some other things, okay, so or if you want to learn some more things, please comment down below, okay, in the comment section. So let me know that what you want to learn next, okay, and I will come up with a video for the same. Okay, so that is all. So don't forget to hit the like button, share the video with your peers, okay, or whoever is in need of this particular thing, or someone wants to upgrade to the bioinformatics field or AI/ML based field. Okay, share with them. I will try to come up with more of simple videos as well. Okay, so this is the first video, so I wanted to tell some things which are already going in the market, okay, and which are basically nailing the market, okay, out there. So you should be versed with all of those things. Okay, so I feel so. Okay, but if you are just starting your career in this, or basically getting started with this particular field, okay, so you might have to take some basics also. Okay, so I will also come up with some of the very basic demonstrations as well, okay, in the future. Okay, so that is all from my side. This is Banerjee signing off. Okay, see you in the next one. Bye bye, take care.